In this video, we're going to discuss DNA replication as part of the second key area of human cells. If you've not yet watched the video on DNA structure, I suggest you start there because if you are comfortable with how DNA is structured, it'll make it much easier to understand how DNA is then replicated. We'll start by talking about primers, as this is what starts replication. We need primers in order for DNA polymerase, the enzyme that actually builds the new strand of DNA, in order to do the whole process. Okay, so this will be your starting point every time we want to make new DNA strands. Okay? So this is all happening just before cell division. Remember, we want to make sure that the new daughter cells have the right number of chromosomes, so we need to start by copying the DNA molecule. And we want to do this faithfully, we don't want to make any mistakes, as we'll see the consequences of that in the mutation section. So we want to make an exact replica or an exact copy of the original DNA template. Okay. So the strands there are going to unwind and separate, allowing a primer, which is a short strand of nucleotides already joined together, which will attach on at the 3' end. So the primer attaches to the DNA template, and that starts the replication process. It marks the spot for the DNA polymerase, so it knows where to start adding in the rest of the nucleotides. So a little bit more about that really important enzyme, DNA polymerase, which is going to build the rest of the DNA strand. Okay. So again, we've broken the hydrogen bonds that were keeping the bases together, separated out the two strands, so each are now acting as a template. So once those primers have attached, we can start extending, start to build up the rest of the strand. Okay. But DNA polymerase, it can only add nucleotides in one direction. New nucleotides can only be joined at the 3' end. So the 3' end of the primer is where DNA polymerase will start adding its new nucleotides. Remember, DNA is double-stranded, it's anti-parallel, which means one end will be 3' and the opposite will be 5'. Okay. That creates a little bit of an issue when it comes to copying the DNA molecule, so it means only one of the strands will have its 3' end exposed and easily accessible to add in the primer and start building in the 5 to 3' direction. Okay. Primer attaches at the 3' end. We then add on the nucleotides at the 3' end of the primer, build up towards the replication fork. Okay. So that's easy for the first strand, it's the 3' end exposed, and the 5' end still zipped, because we can build it in a continuous fashion. One primer, which is continuously extended by DNA polymerase. Okay. Problem becomes for the second strand, which it's got its 5' end exposed and its 3' end here within the zipped area. It's not accessible, so the solution is to add in multiple primers. We put our primer as close as we can to the replication fork, we replicate that primer, and we do so in fragments. Hence the discontinuous nature. Primer, extend the primer, add in the next primer, extend that, next primer, extend in the same fashion until finally you have the whole strand copied. Okay. So that creates a leading strand, it's built continuously, and a lagging strand is built discontinuously into fragments. So the last part of replicating or copying our DNA strand is because we've got fragments of our lagging strand, we've got to then join those together, and we do so by using the enzyme ligase. So ligase will join together those fragments to make sure we've got a complete new strand of DNA. So we've copied both our strands, we've copied the entire DNA molecule, we can now do cell division. So to summarise, first the DNA was unwound, the hydrogen bonds between the bases were broken so the strands could separate. Our primers are pre-attached nucleotides, add themselves onto the 3' end of our DNA template. That gives DNA polymerase its starting point and it can add new nucleotides to the 3' end of those primers and thus extend the leading strand towards the replication fork in a continuous fashion, but building the second strand, the lagging strand, in fragments. Finally, DNA ligase coming along and joining those fragments together to give us our new DNA molecule. One with the original strand, and the second being our newly synthesised strand. So past paper question time. So let's start with this one. Strand B is replicated continuously. Strand A can only be replicated in fragments. So you're being asked to explain why the strands are replicated in these different ways. This is your explanation level question. We've got to provide reasons and scientific explanations for why something is the case. Okay. So pause the video and come up with your response. So let's take a look at the marking scheme for this one. So why the strands are replicated differently, that comes back to this directionality, and because nucleotides can only be added to the 3' end, either in the strand or of the primer. You can see here that nucleotides is underlined. It's a key word that you have to use in your explanation. They will not accept bases because it's the entire nucleoside, the sugar plus the base plus the phosphate that is being added here. Or alternatively, you could have said the DNA strands can only be replicated from the 5 to 3' direction. So it's about this idea that things can only be added to the 3' end, thus replication only happens in one direction. Let's try another, this time about the enzymes that are involved in DNA replication. So you're asked to describe the role of DNA polymerase and DNA ligase. Okay. So again, pause the video, write down your responses. So what you need to include here is for DNA polymerase, this is what that adds nucleotides to the new strand or the new primer. Again, nucleotides has been underlined here. You cannot write bases, you must use the term nucleotide. It's not just the base, it's the full thing. But again, there are still different ways that you could word this. Alternatives that adds, as long as it's clear that it's new nucleotides that's been put on or added here to this new strand or to the primer. And we're building up this new sugar phosphate backbone with the bases in the centre. And for ligase, this is the one that joins the fragments, which was enough. But you could also add either the fragments of DNA or the fragments of the lagging strand. So I hope this helped give you a better appreciation of DNA replication. So start with the structure, comfortable with that, and then think about, well, how do we make a copy of this molecule? Any questions, queries, feel free to leave below.